I'm now going to explain you the structure of skeletal muscle. Skeletal muscle are those muscles that are moving. Of course, your skeleton, so they're responsible for movement. You also have cardiac muscle, which I'm not going to talk about, and you also have smooth muscles, which can be found in your digestive system and in your blood vessels. But these are not the muscles I'm going to talk about. I'm going to talk about skeletal muscle over here. And their second name for skeletal muscle is also known as striated. Striated muscles, and striated muscles are characterized, and you see it over here by these striations. So you see over here that it's bright and light and dark and light and dark, and these are the striations, okay? So if you see a picture that has all these zebra type of pattern, then you know that we're talking about skeletal muscles over here. Now these dark dots over here, that's nucleus, that's another nucleus, that's another nucleus, okay? And what we have over here, this whole thing here, that is I don't know, you might not be able to see it quite well. This must be over here. That's a cell, right? A muscle fiber, okay? The whole thing. And what you see is that in one muscle fiber, you have multiple nuclei, okay? So that if you remember the so-called exceptions to the cell theory in chapter 1, it mentions that the cells are fused together into long fibers and that there are multiple nuclei. So that is the structure of a skeletal muscle, and what we're doing right now is I'm zooming into these parts over here to explain to you how the muscle contracts, okay? Because it's able to pull together and is able to contract, and we're going to now really zoom in and we're going to have a look at this in more detail. But please remember, you've got to be able to identify skeletal muscles. You have to know it's also called striated, okay? And you have all these alternating light and dark bands. They're called light and dark bands. Okay, so let's move on. So what you see over here is you now see the zoomed in version over here, okay? And I'm going to explain you now what this is here. This from over here to over here, this is the dark band, and from over here to approximately over here, that's the light band. So you write down dark, that's the light band. Okay, and please notice that there is this really dark line over here, right? Okay, this real dark line. This dark line is known as the Z line. You also see it over here, right? That's the one over here. But this Z line is part of the light band. Okay, please, that's kind of confusing sometimes for some people. How can it be that something which is very dark, almost black, like over here, how can this be part of the light band? Yeah, it's very thin, okay? It's part of the light band, yeah? And the structural or the contractile unit of a muscle is called the sarcomere. That is also an important word. And the sarcomere goes from the Z line to the next Z line. So that is from over here to over here, okay? That is one contractile unit, okay? Now, how does this work? There are two types of protein filaments or protein fibers involved here. Okay, you see over here these fibers over here, they are called the thick filaments. Okay, here in blue, the thick filaments, that's myosin. You have to know this as well. Myosin, that is a protein, and that is the name of the protein of the thick filaments. Okay, and that's the myosin. And you have thin filaments over here. Okay, these ones over here. Okay, and these thin filaments, they also have a name. They're called actin. Actin and myosin, okay? And what happens now is okay, let's just label a few other things over here. Some people call this the A band over here, where you only see the myosin, okay? The I band over here, where you only have the actin, okay? And you can also see that the A and the I band, they refer to the dark and to the light band, okay? So in other words, we have actin and myosin filaments, and what you see is that there are next to each other. And how can it contract? They can contract because the actin and myosin, they slide past each other. So the filaments, they stay the same length, okay? They stay in the same length. The filaments, okay? Actin, myosin, yeah, they stay the same length during muscle contraction, but what they do is they slide past each other, okay? And this is how it is contracted by sliding past. Okay, so one thing is you gotta be, you just remember and you study this whole thing, and then the structure and all the parts, and then of course over here you have a new sarcomere starting and then another one and so on. Okay, so you have always these light and the dark bands alternating. Yeah, and these filaments, they slide past each other, and I'm going to show you in the next foil how this actually happens. So what you see over here is, again, you see the light and the dark bands. So that is over here, that's the Z line. Okay, that is the light band. It only has actin. Okay, that is the dark band. It only has myosin. I mean, the actin fibers, the actin filaments, they go from the light band and also extend into here, of course. But what you have over here then is you have the myosin going here, right? Okay, and then of course you have so that's the dark band, and then you have again a light band, and the dark band, and so on. Okay, you get the idea. Okay, and this muscle over here, that's relaxed. Okay, and one sarcomere goes from here to here. It's a sarcomere. Sarcomere from Z line to Z line. Okay, and now the thing is that this one is relaxed. And in the relaxed state, you see that we have a large light band. Okay, so I don't know how am I going to write this down here. Relaxed. Okay, the light band is large. Okay, and down here we have a contracted state, and you see that the myosin did not change. That's contracted. Okay, and what happens is the actin and the myosins were sliding past each other, but the myosin did not change in length, but the light band is narrow. Okay, so you see over here, yeah, the light band is, yeah, but the dark band did not change. That is important. Okay, the dark band, that's the myosin did not change in length. Okay, it's simply that the light band got more narrow, and so the overall sarcomere still from Z line to Z line over here, it has also contracted now, of course. Okay, so the bottom line is just make sure that when the muscle is contracted, then the light band is small. When it is relaxed, the light band is large. The dark band does not change, okay? Because the myosin does not become shorter. The myosin makes it dark, it does not change, okay? Now, let's have a yet further detailed picture over here. And you see it now how it actually looks like. Well, it's still only a drawing over here. What you have is you have the over here the relaxed state and the contracted state. It's not as extreme as in my previous picture, but what you see here is that you see the myosin here, and you see that these myosins, they have these little heads over here. They look a little bit like golf clubs, okay, sticking out. And these little heads over here, they are able to cross-link with the actin, because that's of course the actin, okay. The red one here, that is the myosin, okay. And these little myosins, they have heads, and these heads they cross-link with the actin, and they're able to move back, they flip back, and that's how it's able to contract. And I'm going to explain in a different foil how this happens on a chemical level. Okay, but that is simply so the idea here is that you have to understand that the myosins over here, there's actually a label over here. You see myosin, that the myosin has a so-called head. Over here they call it the Z disc. I call it the Z line. That's the same thing. Okay, you don't have to know this one over here, the cap Z. You don't have to know the myosin tail. Gee, you don't have to know that either. Okay. So what you have to know, of course, you gotta know the actin, you gotta know, you gotta know the myosin, of course. That is really important. Yeah, you have to be able to identify the Z line. That's the Z line over here. Okay, yeah, and of course the light and the dark band. Now, where is the light band over here? Well, the light band is there where you don't have any myosin. So the light band goes from here to the next one, because it goes on here as well, right? The next sarcomere starts, right? And the dark band is where the myosin is. So that's the dark band over here, from myosin to myosin. Okay, and what you see over here is in the contracted state again, is you see that the light band becomes more narrow, yeah, because that's where you don't have any myosin. So okay, and they call this the M line. I don't know. Yeah, I don't know if this is so important or not. Yeah, just remember the M line is in the middle of the myosin. Okay, yeah, No, that's a bad drawing. Okay. So that's it. Okay. So that is simply the overview now, and in a different video I'm going to explain now how this actually happens. Where do you need ATP, for example? The whole thing, muscle contraction, needs energy, and the energy is used to flip the head of the myosin back. And how does this work? Well, this is I'm going to explain in a different video. Okay. So all the best.